the central dogma of biology. That sounds profound, right? The central dogma of biology. So, has anybody ever seen that term before? Dogma? What does dogma mean? What's dogma mean? Anybody know? It's like a belief system, a set of beliefs, right? And so the central dogma of biology is like, this is like the main theory, the main thing that we're going to base all of biology on, and we can explain things away using just this theory. Okay, and this theory goes like this. DNA is used to make RNA, and then RNA is used to make proteins. It's as simple as that. Organisms have DNA, that DNA is used to make RNA, the RNA is used to make proteins. The proteins go on and do stuff. Mutations in the DNA causes different RNA to be made, which causes different proteins to be made. Those proteins do unique things. Then we've got new mutations. Like that's the whole idea behind how there is diversity in life. Okay, so this process of using DNA to create RNA is called transcription. That says that RNA can be created using a DNA template, and it's going to require an enzyme to do that. The enzyme that does it is called RNA polymerase. Okay. Then once you've got RNA, we know we can go and make proteins then. That process is called translation. Translation says that proteins are produced when the nucleic acid sequence on mRNA is translated into an amino acid sequence. And the enzymes that are involved in that, or the things that are involved in that, are tRNA, and ribosomes. Okay, so the actual enzymes are the ones that are in that ribosome, the ribozymes, if you will. Okay? So there's a problem, though, and the problem is that the central dogma says you can make DNA, or you can make RNA using DNA, and you can make proteins using RNA, but it never talks about making DNA. And we know you have to make DNA, right? And so the one thing that it doesn't say is that DNA can make more DNA. Okay, and that process is called replication. And it says that DNA can make an exact copy of itself. Okay, and we're not going to list all the enzymes involved here. We're just going to say many enzymes are involved. Okay, and that's what we're going to spend our time talking about today, is replication. Okay, so let's go talk about replication. First thing that we need to know is a little bit about DNA. Okay, so here's a long DNA strand, okay, one strand of the double helix. Then we've got the other strand. I'll do it real big down here. Okay, and so yeah, that's right. On this, you've got nucleotides. And you may have read last night also about the way that we talk about the direction of DNA, okay? And that is by using the terms 3' and 5'. And that's a term that comes from organic chemistry because we know that DNA has a 5-carbon sugar in it. It's got deoxyribose, which is a pentose in it. And so because it has 5 carbons in that sugar, we name the carbons carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5, right? And so by saying which end is exposed in order to bond to, we can tell which direction the DNA is going. So on one side, we could say that this is like the 3' end, meaning over here, there is a 3 carbon that's open for bonding. And over here, we'd say this is the 5' end. And this says that the 5 carbon is open for bonding over here. Okay? So the other strand then is going to run anti-parallel, which means that it's going to be the opposite. So if the 5 carbon chain, or if the 5' carbon is over here, then the 3' carbon is over here, then the 5' carbon will be on the bottom. Like that. Okay? So anti-parallel in opposite directions from one another. You don't have to write this, but you remember how when we did the molecule of DNA, when we did a single nucleotide, we said, okay, you got like the phosphate group and then the sugar and then the nitrogenous base, and we sort of drew it like that. And then we said that there would be like another phosphate group here, another sugar, and another nitrogen or nitrogenous base, right? See how they're sort of like this? They kind of look like little L's. Well, you can think of this anti-parallel thing as having a bunch of L's that are going this way, and then a bunch that are going this way. Right? You see how they're like opposite of one another, anti-parallel, right? Okay, so if we know the nucleotides here, let's say we've got A, C, T, T, G, C, T. Okay, then we know they're complementary base pairs. T, G, A, C, G, A. Right, because A is always going to be complementary base paired with T, and G is always going to be with C, right? So these are held together by hydrogen bonding most of the time, and if we were to unhydrogen bond them together, we were just able to sort of pull them apart a little bit, then nucleotides would automatically just kind of come in here and attach, right? They would hydrogen bond to these open positions then. So if I'm able to separate my 2 strands of DNA, then the next logical thing that happens is that the nucleotide bases will come in here and complementary base pair. Like this. So now, if you look, I actually have 2 identical strands of DNA that are made because I split my DNA in half, right? I pulled the 2 molecules of DNA apart, and then I have now created an exact copy of the top half on the bottom, and I've created an exact copy of the bottom half on the top, right? Okay. So then all I need to do now is find a way to connect these nucleotides together. So an enzyme, specifically the enzyme DNA polymerase, is going to come along and it's going to connect all the sugar phosphate backbones together, like so. And now I have 2 identical strands of DNA, right? And again, these are anti-parallel again, so the 5' end is going to be there, and the 3' end is there, and this was the 3' end, and this is the 5' end. Okay? This process, like I said, is called DNA replication, Okay, and we can say that we know that after replication, each double helix contains one original strand, which we've drawn in black, and one new strand, which we've drawn in blue. Okay. For this reason, the process of DNA replication is called semi-conservative, which means it's not destroying the original strands of DNA when this is happening. It's keeping them. And so as a cell line continues, it continues to use more and more and more of that DNA. Or continues to use that DNA over and over again, rather. It's for this reason that it's theoretically possible, it's wildly unlikely, but theoretically possible that you have in your body right now one of the original strands of DNA that was in the sperm or the egg that created the zygote that you came from, right? Theoretically, that's possible. If it was a long-lasting cell like a nerve cell, right, it's possible it could be from the original germline and then still have that original strand of DNA in it from the sperm or the egg, which I think is mind-blowing to think about, that it's lasted this long, and that it's still there functioning and doing its job. Okay, again, it's unlikely, but still cool to think about. All right, so let's talk about how this process actually works, how replication actually works. 1, 2, 3, sure, there and there. So I'm going to draw in a weird angle to draw this. Sure, that's fine. And then this 1, 2, 3. Nope, messed that up, huh? This one was supposed to go up to here. Sure. Sort of like a little bubble, right? Okay, so on this side, the base pairs are hydrogen bonded together, and on that side, the base pairs are hydrogen bonded together. This structure that we've drawn here is called a replication bubble. Okay, and during the process of replication, hundreds of replication bubbles will open up on every molecule of DNA, on every DNA strand, and they will, every DNA double helix rather, and they will simultaneously start replicating the DNA. All right. So in between here are all the unzipped or unwound nucleotides that are now exposed. Okay, so question number 1 that needs to be answered: how did this DNA double helix get unwound? And the answer to that is there is a enzyme that does it. Okay, that travels in this direction, we'll say. And this enzyme is called DNA helicase. Okay, and the job of DNA helicase is to go down the line and separate the double helix, or unwind the double helix. So we'll say that DNA helicase, that is the enzyme that unzips the double helix at the replication fork. Okay? So this area where it's actively unzipping it, that area is then called the replication fork. Okay, so up here, this is the replication fork. And that's always on the opposite side of this area, which is called the origin of replication. So one side is the replication fork that's actively opening, and the other side is called the origin of replication that is not actively opening. This side stays closed. Okay? For the purpose of this discussion, let's label the two strands. So this is going to be our 3' end, and this is our 5' end. And then over here, that makes this the 5' end, and this the 3' end. So the first thing that has to happen is actually a little bit confusing, and that is that DNA molecules cannot just start making themselves from scratch. Okay? They can't connect together if they don't have anything to connect to. And so in order to get this new strand going, the first thing that has to be added is a primer. So this is an RNA primer. So a couple of RNA nucleotides get added on, not DNA nucleotides, RNA nucleotides. They get added on to be able to start this new strand. Okay? So there's obviously going to need to be an enzyme that does this, and the enzyme that does this is called DNA primase. That makes sense, right? So this adds a short RNA primer in order to start a new strand of DNA. Okay. So there's a primer there. There's also going to be a primer here. Now the reason that I've put them here in these locations is that the new strand then has a 5' end and it has a 3' end, right? And it's anti-parallel. So if the 3' end is here, the 5' end has to be anti-parallel. So that means that the 3' end of the new strand is right here, right? So that means that this one is going to be creating a new strand in this direction, and this one's going to be creating a new strand in this direction. Right? So as you unzip the helix, as I said, new nucleotides are just going to start sort of attaching here. These are new DNA nucleotides. Same thing down here. But these DNA nucleotides, they are not attached together yet. They can complementary base pair, because complementary base pairing uses hydrogen bonding, and hydrogen bonding is not a bond, it's an intermolecular force. And so we know that it doesn't require any enzymes to do that. But if they want to connect the sugar phosphate backbones together, remember, because all nucleotides have a sugar and a phosphate on them, and if they want to covalently bond those together, there's going to need to be an enzyme there to do that. Okay, and sure enough, the enzyme that's going to do that will travel along in the same direction that the strand is being made, and this enzyme is called DNA polymerase. Which makes total sense when you think about it, because the enzyme's called DNA polymerase, and it's the thing that's making a DNA polymer. right? It's joining the molecules together. So, next one is called DNA polymerase. So this is the enzyme that joins the sugar phosphate backbone together. At the 3' end of the growing strand. But next we're going to have a little bit of a problem. Okay? Here's the problem. When we go up here, this top strand is moving in this direction. Like that. Okay? So when it gets to over here, what is the DNA helicase going to do for it? It's going to open up more double helix, right? So it can continue to go in this direction. Right? What about here? There's no way to open it up because there's no helicase over there. All right? So this bottom strand, what it does is it starts out right next to the DNA helicase, and it makes however long of a strand it can, and in the time that it takes to make this strand, this DNA helicase will have moved on further in this direction. Okay? So what it's going to do is it's going to move it this way, and then it's going to jump back to the next spot where the DNA helicase is, and it's going to make that fragment, and then it's going to jump back again and make another fragment, and another fragment, and another fragment. So the top strand is going to be one long chain, one long strand, and the bottom strand is going to be a whole bunch of little fragments. Right? So we have a name for this. The top strand is called the leading strand. And the bottom strand is called the lagging strand. And that's actually not the best terms for it because the lagging strand doesn't go any slower than the leading strand, even though it sounds like it. It just happens to be that it gets copied like slightly behind the leading strand because it's not going to be directly next to the replication fork. And I'll show you the actual complex. There's a really good animation of the actual complex of all these enzymes that's doing all of this replication, and it'll make more sense when I do that. But for now, just know leading strand and lagging strand. Okay? So this is going to cause, like I said, a problem. We're going to have a whole bunch of little fragments in the bottom here. And so we have to have an enzyme that will connect those fragments together. That enzyme is called DNAligase. And it uses this prefix lig. Does anybody know what it means to ligate something? Does anybody know what a ligament is? It's a connection between bones, right? So that means that if this is using the same prefix, what it means is that it's connecting these fragments together, right? So this is the enzyme that connects the fragments in the lagging strand. Okay, the term for these fragments that are created in the lagging strand is called Okazaki fragments.
Teachers and students, if I might have your attention, please. We will be holding the bell. Please do not release anybody for lunch until the bell rings. Thank you.
So these Okazaki fragments, these are obviously named for the scientists that discover them, the biologists that discover them, but they are the individual little parts of the lagging strand. Okay? The last enzyme that we're going to talk about is called DNA Topoisomerase. So all the other ones mean exactly what they do, or are named for exactly what they do. This one is not so much. Maybe it is, and I just can't decode it well enough. But I don't think that it is. Okay, so DNA topoisomerase's job is to prevent the overwinding of DNA. And the way that it does that is it cuts the DNA to release tension and then joins it back together. So let's talk about this for a second. Let's say you pull your headphones out of your pocket and they're all tangled up. It's a big tangled mess. Okay. The smart way to do it is to find an end, like the plug, and just kind of snake it back through until it's untangled. Right. The dumb way to do it is to just take two random pieces of wire and then just start pulling. Right. And be like, it'll eventually come out if I pull hard enough. Right. Well, the enzyme helicase is dumb. It doesn't know how to do anything else. So what it's going to do is it's going to take this double helix that's wound up, right? And it's going to take two pieces of it, and it's just going to start pulling, right? And if you've ever taken a piece of string or something like that and tried to pull out the winding part of it, at one end you get a really big knot, right? Because it gets even more tightly wound. And so if you continue to unwind the double helix without letting it sort of spin its way out, then it will eventually get all knotted up over here and break. Okay, and if it breaks, that won't be a clean break. And if it's not a clean break, there's no way for enzymes to put it back together. You will have destroyed the DNA at that point. That's a big problem. So what happens instead is that there's an enzyme over here called topoisomerase, and every once in a while it chops the DNA and lets it sort of spin out that extra bit of tangledness of winding that it had, and then it connects it back together so that it doesn't get overly wound up and cause problems. Anybody have questions on replication? Yeah?
Do different species have like the different lengths of chromosomes because of that?
What do you mean?
Like, or like is DNA like different lengths because of the
Topoisomerase? No. Interesting thing about this, though, is because of the way that Okazaki fragments work, and because the fact that you have to put an RNA primer on it to get it started, every time that DNA is replicated, you lose a couple of nucleotides off the end. And if you remember from I think we've talked about it a couple of times, there are special sections on the end of each chromosome called telomeres, and those telomeres are non-coding DNA, and they are non-coding DNA because every time it gets replicated, the telomere gets shorter and shorter and shorter, and once the telomere reaches a certain size, then that can't be replicated anymore, and that's the end of that cell line. It can't replicate anymore, which is why I guess if they're holding the bell, I'll talk. There's this whenever we do human research on human cells, we want to make sure something is going to act a certain way. We use a specific human cell from one person. Okay, there's this lady named Henrietta Lacks, and Henrietta Lacks had cancer, right? And she had what was called immortal cancer. She had a cancer that had turned back on the gene that allows her to create new telomeres, right? And so her cell line is immortal. You can culture it and it will continue to grow. It's a cancerous human cell, but it continues to grow forever. So basically every new drug that gets tested, every single study that's ever been done in the last 40 years uses these same cells called HeLa cells, H-E-L-A, and they're named for Henrietta Lacks, who was the original person who had cancer that all these immortal cells came from. And the interesting thing about it is she was never asked if we could use her cells. Her doctor had taken a sample to biopsy and noticed that they were immortal, and so he just kind of kept them, and then she died, and then he just kind of kept using them, and then they were like, well, no harm done now. Let's just keep using them because she's dead. But there's a really good book out called The Secret of Henrietta Lacks or something like that is really good. Yeah?
Can I see the definition of DNA?
What's up?
Oh, I was gonna wonder if she died, or like how she died.
She died of cancer, yeah. Of the cancer.
How would that come
It started taking over other organ systems in her body. It metastasized. Right? So it was a type of she had cancer, right? And it just happened to be that one of the weird mutations that her cancer cells had was that they could divide and replicate indefinitely. Whereas normal cells, most cells, will divide a bunch of times. Even cancerous cells will divide a bunch of times, and then they start destroying their own DNA because there's no more telomeres on them. And so then eventually they'll kill themselves off. Normally, they kill you before they kill themselves off, but they will eventually do that because they'll run out of telomeres. Yeah. A little bit up? Sure. Anything else? There's also another really good documentary called The Mystery of Photo 51, which is about what you guys read about last night, which is the X-ray crystallography and how Watson and Crick discovered that the DNA was a double helix. They had this X-ray crystallography photo that was actually not theirs, and a fellow researcher had left it on her desk. That she was doing this research on X-ray crystallography and happened to do DNA, and Watson just kind of like walked by it and was like, I know what that is. I've seen that shape before. And he picked up the photo and took it with him and never credited her. Her name was Rosalind Franklin. And when Watson and Crick won the Nobel Prize, it was big controversy because they said that Rosalind Franklin's name should have been on it as well, even though she was dead at the time. But she never got credited. There's a really good Discovery Channel special. I think it's a Nova special, actually, on that. Any questions? They gave me time, so I talked.